Welcome to the first of two New England Journal of Medicine special podcasts on avian influenza. Today we'll be focusing on the biology of the disease and its potential to result in a human outbreak. I'm Stephen Morrissey, managing editor of the journal, and I'm talking with Eric Rubin, editor-in-chief, and Lindsey Baden, deputy editor. And we have two guests to help us understand where we are now. Arnold Monto is the Thomas Francis Jr. Collegiate Professor Emeritus of Public Health at the University of Michigan. He has a long history of studying the epidemiology of influenza and the interventions to prevent and treat the disease. Jeanne Marrazzo is the director of the National Institute of Allergy and Infectious Diseases at the NIH. In that position, she has a vital role in coordinating the research that supports our understanding of influenza and that can lead to new approaches to preventing and treating disease. Before we start, Eric, could you help our listeners understand why we've decided to record these podcasts?
Thanks, Steve. Of course, avian flu is an important issue. I think it's important because most infectious disease experts have been waiting for a large pandemic to occur. It came, but it was actually a coronavirus. However, we've all been suspecting that there will be a large outbreak of influenza. It's important to note, however, that this current avian flu outbreak isn't an outbreak at all, at least among humans. So far, we only know of one person who was infected and developed disease, and that case was fortunately relatively mild. Nevertheless, the current strain of flu that's circulating among cattle in the US has acquired at least some of the mutations that we expect to be necessary to transmit well in some mammals. Hopefully, this one won't acquire any more mutations. But even if this particular strain doesn't do so, we suspect that eventually some influenza strain will be able to make the jump from animals to humans and to transmit efficiently. So we thought this was a good opportunity to talk about flu and its potential.
Arnold, let's start by following up on that. What exactly is avian flu, and what characteristics does an influenza strain have to become easily transmissible in humans?
Well, the original hosts of type A influenza, which is what we were talking about, is waterfowl and other free-ranging avian hosts, shorebirds, etc. They are the ones that have influenza and continually spread influenza in the wild. We get infected from influenza by other variants of type A, which have developed the characteristics of spreading from person to person. Basically, receptors for influenza for the avian strains are different from the human strains, and that's what more or less has kept them separate. And what we're always worried about, because avian strains are in a great deal of variation. There are a lot of varieties of avian strains and only 3 subtypes that regularly infect humans. One of them hasn't been seen for more than around 50 years or so. So we deal with 2 subtypes, AH3N2 and AH1N1 of type A influenza, but out there in the wild, there are 16, 17 different subtypes. And some of them are quite lethal in birds, and that's the concern.
Arnold, you mentioned that the receptor, whether it's avian receptor versus mammalian receptor, is an important sort of species defense. My understanding, correct me if I'm wrong, the flu genome is 8 segments and it can recombinate. And that's one of the ways it gains potential pathogenicity. This particular strain that's circulating in the dairy cattle has the PB2 gene that seems mammalian adapted. What does that mean? Is that a concerning feature?
Well, it's a concerning feature, but we've seen variations like this with the H5N1 virus. Now, the H5N1 virus first infected humans in 1997 to everybody's great surprise, because avian strains, the high pathogenic avian strains, were not supposed to infect humans. And there was an outbreak in Hong Kong, and people died from it. People thought that this was a one-off. And then in the fateful year of 2003, when we saw SARS, we also began to see cases of H5N1 coming from wild birds into humans. And there was a widespread alert because there were a few cases occurring in humans. And we developed in the United States and elsewhere the kind of non-pharmaceutical interventions that were rolled out during the COVID pandemic. And there was real concern because it was lethal when it got into humans that it might change enough antigenically based on segment changes in the genome to become adapted to humans. It's never happened. But in the process, we have learned that a lot of mammals can be infected by this avian influenza viruses, but not dairy cattle, not cattle, I should say. And that's the surprise here.
So, Jeanne, the NIH coordinates with other federal agencies, particularly the CDC, to monitor outbreaks. Is there a similar mechanism for following disease in animals?
Yeah, Steve, that's a great question. We have been in this current outbreak working very closely with the USDA, with the Assistant Secretary for Pandemic Preparedness, and with the CDC to try to look at this. Obviously, the USDA is the one that's really responsible for monitoring disease in animals in domestic situations like we're talking about, such as farms. But what we do in specifically that I think is worth mentioning and has proved to be a big foot up in this outbreak investigation is to fund a group called the CEERS, which is the Centers of Excellence for Influenza Research and Response. The reason this group is key is not just because it has very good relationships with many farms and many state veterinarians, because they have been doing active surveillance in some of these places. They've also been very involved in doing wildlife surveillance for many years. And you may remember that people have been increasingly concerned in the last several years about massive sea mammal die-offs. One of the big questions, we knew, as Dr. Monto said, that wild birds were going to be a problem, increasingly a big reservoir, more species, more spread, cross-seasonal occurrence as opposed to just sort of more sort of migrational patterns. But when we started to see sea lions, in particular, there's a huge die-off in Maine a couple of years ago. We now know that harbor seals in Argentina have just thousands and thousands of animals have died. So CEER has been sampling those mammals for many years. So since 2014, they've actually sampled about 18,000 marine mammals, in addition to kind of the more mundane wildlife, but bird life, wild bird life sampling. So it's interesting because marine mammals, because they interact closely with the birds around predation, but also because of habitat overlap that is probably changing in part because of climate change. All of this is creating, I think, an opportunity for some really rich signals, and clearly we have been seeing them. So those are just some of the things that they have been doing and have been able to sort of, I think, see this coming in a way that certainly predicted more interspecies transmission, perhaps not yet humans, but again, very concerning. I wanted to come back to one question that Lindsey asked, and that was this issue of the cows getting infected. There was a very interesting preprint you may have seen recently that looked at those sialic acid receptors in the mammary gland of cows and showed that not only were there the duct receptors, but there were also human receptors. And so the theory was that actually the mammary gland, for reasons that we had not predicted at all, would be more of an opportunity for sort of rich mixing of these genetic populations of virus. So very surprising, and may explain why we're seeing such high levels of this virus in those cows and in the milk in particular.
Jeannie, in thinking about that, that sort of raises the issue of how we think about our level of concern for the emergence of strains that could efficiently transmit between people, and changing the binding receptor from, the 2,3 to the 2,6 sialic acid is something which we're concerned about. We saw in 2009 with H1N1 pandemic that it was in swine that mixing occurred. Should we be concerned if it jumps to swine from spillover, or perhaps from the observation you just mentioned, this concern may already be going on in the mammary glands? How can we be thinking about this issue?
So a couple things. Remember that the conjunctivitis case in this current description, or this current quote unquote outbreak, in some ways is not new. There was an outbreak, a large outbreak of conjunctivitis in the Netherlands about 20 years ago now or so. Remember that you've got alpha 2,3 sialic acid receptors in the conjunctiva. So conjunctiva technically is more receptive to these avian flu strains. So I think the question is that going to be enough? No, I think other things are going to need to happen to make this particular virus, as Dr. Monto said, much more invasive for us. And that's going to involve probably not only sort of a more efficient binding through those sialic acid receptors, but additional changes in the polymerase, the PB2 gene. You mentioned the human case had one change that is pretty typical of mammalian adaptation, but I think you're probably going to need some more for it to really efficiently get into the respiratory tract. The other thing is, some people think that it's going to need to mutate more so that it is more efficiently transmitted in air droplets. There are some changes that happen to the conformation of the virus under shifting pH, and if that is modified at all, you may have more trouble transmitting it through aerosol. So I think those are a few things, and then we can't, of course, forget the hemagglutinin changes that I mentioned regarding carbohydrates. So it's possible that the mammary gland may provide more of that opportunity. What I worry about a lot that I don't think people are talking very much about are the peri-domestic animals on the farms. So if you read that emerging infectious disease paper that came out last week, they did a detailed analysis of at least one necropsy of a cat. The cat had very impressive central nervous system infection, disseminated infection. Number of cats on farms have clearly had clinical encephalitis. A number of cats have died. So this concept that you're going to keep pigs separate on the farms from cows separate on the farms from the humans who go between them, I worry very much that with the animals like the cats, possibly the dogs, whoever else running around, that's going to be very tough to do.
Since we're talking about animals, there have been very large animal outbreaks of various influenza viruses, largely in China, for years and years and years. And when we've seen movement between animals and humans, we've seen it in China. There hasn't been a sustained outbreak in humans yet, fortunately. But it's interesting that we're seeing this one in the US, which doesn't have the same concentration of domestic chickens and ducks that were always the sort of nidus of infection in China. I know you can't answer why, but why? Any idea what's going on that's different this time that's allowing a U.S. outbreak to occur?
I can give you one thought. I would love to hear Dr. Monto's thoughts. I mean, I've learned a lot about how cows are moved around the country. So it's not sort of the pastoral view that you have on the organic milk cartons of the little cow that stays in its pasture and is milked by the little children. In fact, cows are raised in a certain area to get to the point where they are milkable or likely to produce milk, they're often moved to large milking facilities, right, with a lot of transmission potential through infection at the udders because the milking parlors are big. There's a lot of milk that goes on the floor. They use very high pressure hoses to wash the floor, which presents the opportunity for significant aerosolization. So it's not quite as controlled an environment. And then the cows are often shipped back to other places for various other parts of their season or their lives. So when you look at the spread of many of these cows that have been detected in these herds, many of them have come from cows that probably were originally infected in the initial Texas outbreak. So you can trace them back. So it's hard to contrast that with what goes on in China. I don't know if that's a similar situation, but looking at that interstate transmission and all of that kind of mobility opens up a whole new dimension of how this thing could be moving around more efficiently.
So, as you say, H5N1 has been circulating in animals, including domestic animals, for a long time. What's prevented it from making the jump to becoming a more humanized virus?
You know, that's a very good question because one of the issues we have now is, and we've had this with H5N1 ever since the 2003 beginning of recognition of how widespread the cases have been from time to time in humans. And that is, it hasn't made it into humans for all this time, even though we have regular human exposure. Last year, we had a big uptick in highly pathogenic H5N1 in laying hens, which made the egg supply almost disappear in many parts of the country. And yet, with not ideal containment facilities, when they depopulated the farms where the laying hens were, there were no human cases. This is an unusual situation. And the problem here is the danger, if there is a spread because of molecular changes, is very high. The probability is probably very low. And the trick is to balance this out in terms of the response. We know, for example, that there are human cases with deaths occurring in Cambodia. That's been going on for a while. But the concern, and I agreed with Dr. Marrazzo, the dairy cow situation is very concerning. And at the end of the preprint, which came from a SEER investigator, among others, the issue of the mixing vessel is brought up. And we've always worried about the swine as mixing vessels because the pigs have alpha 2,3 and alpha 2,6 receptors. And the question was raised, well, maybe dairy cows could be a mixing vessel because they do have 2,6 receptors with 2,3 receptors in the udders and other receptors in the respiratory tract. And cats are well known to be very susceptible to this virus. And that's happened in cats on the dairy farm. And I think we need to go back and re-examine some of the episodes that occurred during the first scare with this virus. And that is that there were die-offs in zoo tigers in Thailand during that period. And there are a lot of things that we can learn by looking back, at least in terms of where we need to be most concerned in terms of surveillance. We need better surveillance than we have because of a lot of bureaucratic issues of dealing between the avian, the agriculture side and the health side. And we really need to know better what's going on out there.
Just one nomenclature clarification. Highly pathogenic is highly pathogenic to the avian population.
Correct.
And that increases our concern that it might have pathogenicity to mammalian population, but the terminology is really about the avian infection.
Correct.
I wanted to say one other quick thing too, related to our previous assertion, that we have one documented case, which was very impressive conjunctivitis. It was great to see that, by the way, in the journal. There is a fair amount of concern, of course, that we are underestimating what the clinical burden of disease is. And a lot of that really does have to do with the people that we're talking about who are working on the farms, many of whom, may not have easy access to or insured health care, to say the least, and maybe have disincentives even for presenting for care, given concerns about job loss and economic damages. So I think we have to be really sensitive. I think it's encouraging that we're not seeing clinical pockets of conjunctivitis, for example, or febrile illness in some of these places, particularly in areas where we know there are infected herds. But I would not be totally sanguine yet, and I think we have to be really really open. Secretary Becerra did a nice message last week, sort of talking to or directed to many of the farm workers, many of whom are migrant workers, sort of encouraging people to be sure that they are attentive to their health and seek care if they're having any issues. So I think it's an important one. The wastewater data, again, that's coming out is quite interesting. It indicates there's quite a bit of influenza A in the wastewater. In some areas where we don't have documented herd infections. So there's a lot to tease out here yet. And I think this is just the beginning of the story. Hopefully it will not evolve more genetically. But there's a lot more to learn, I think, in terms of the epidemiology of what's going on here.
And we know it's in the milk.
Sure is in the milk. Yes. Yes.
Well, speaking of the end of this, at least for now, it's May. It's a seasonal virus. What do we expect will happen to an outbreak in cattle where we know it's a seasonal virus in humans?
State fair season's coming up, right? If you've lived in a rural area, people go to the state fairs. There's all kinds of cute bovine animals. There's other animals running around. There's kids petting everything, kissing everything. So the worst case scenario is that the seasonal concept does not pertain, which it seems to be what's happening with the wild birds, right? Again, I mentioned that they're just, they seem to be getting more sort of persistently infected. So that's kind of another nightmare scenario. I don't want to be too dramatic. I don't know that seasonal kind of patterns would pertain to this, and I would love to hear Albert's thoughts.
And we know that every summer we have swine influenza viruses transmitting to humans, and it's because of the state farm meetings, the the county fairs, and all the rest.
Yeah, just another excellent interface for human multiple species interactions. So I did want to comment on the milk, though. I think one thing we can say, please don't drink raw milk at this point. I know that there are still many people who believe that, you get a lot of benefit from raw milk, especially if people are immunocompromised, especially for young kids, pregnant women. There are lots of good reasons from, brucellosis to all the other things we think about not to drink raw milk. But given the viral loads in this situation and the fact that, the cats probably drank the milk, so there is probably nasopharyngeal infection based on ingesting the milk just by virtue of the anatomy of the respiratory tract. So I think people really need to be cautious and think twice before either drinking raw milk or giving raw milk to certainly their kids or immunocompromised people.
And we really haven't heard how much raw milk is being licensed and consumed in certain states where it's still legal to sell raw milk.
Yeah, and absolutely. I mean, the estimates that I've seen from the USDA is that maybe 2% of people drink raw milk. That's a lot of people. Plus, there are culturally groups of people who really do drink a lot of raw milk, a lot of raw cheese, in particular. So I think it could be more of a problem than we realize.
Does pasteurization render the milk safe?
So starting with the initial research that the Sears folks teams did, and then much more methodical research by the FDA, which is still ongoing, I think it's pretty safe to say that pasteurization does kill it. It seems to be very effective. I think we have to wait and see what's going on with beef cattle. We haven't mentioned that yet, right? Again, getting to this whole issue of dairy cattle, beef cattle, small farms mixing. Probably going to look to get some data on that soon too, I hope.
So we know a lot about vaccines for influenza. And in fact, there is an H5N1 vaccine on the shelf that hasn't been commercially produced yet. But we know from our current vaccines that small protein sequence variations in each viral strain can have a big impact on vaccine efficacy. So what do we know about how well the vaccine we have matches the strain that's currently infecting cattle?
I'm not quite sure about the current one, but a lot of work has been done by NIAID and other groups. Again, because of the concern 20 years ago or so about spread. And that's why we have H5 vaccines in our stockpile. We've had studies about antigen sparing and all sorts of things using adjuvants. We're also in the new era of mRNA, and I haven't as yet seen very much in terms of a monovalent H5 vaccine. We've had experimental vaccines developed which contain multiple hemagglutinins, including H5. But I think that's something we really need to be following. Part of the problem with a pandemic is you have to respond quickly. And you've got to go what's on the shelf initially. We were very lucky to get a vaccine in under a year with COVID. We may not be lucky again with another pathogen. With flu, it usually takes a while. And the 2009 pandemic, we were fortunate because FDA could use just strain replacement, and we didn't need an adjuvant. Although adjuvanted vaccines were used in Europe for administrative reasons more than scientific reasons.
Yeah, I agree with Dr. Monto. I mean, I think, we do have those two candidate virus vaccines that are, I think, the Astrakhan and the American Widgeon vaccines. And the CDC has reported good cross-reactivity using ferret antisera raised to those two candidate vaccine viruses with pretty good geometric mean titers. So I think that's encouraging, and that's from Vivian Dugan, who leads the influenza group at CDC. We at NIAID and our investigators have looked at high titer sera samples from people immunized with an H5N8 strain, and that has shown pretty good substantial cross-reactive antibody titers to this bovine strain. But I think Dr. Monto is right. We got to keep digging in here and looking and making sure that we're continuing to track this down. The other thing we're really involved in, although it's in very early stages, and as Lindsey knows in particular, it's in an area that we are very invested in, is looking for monoclonal antibodies, especially in sera from people who've been vaccinated to those H5N1 strains, to try to isolate what the most potent either monoclonal or even broadly neutralizing antibodies might be. So that's been gearing up. Again, it's going to be a little bit more time, of course, than what we've got on the shelf. But I think it is important to go down that road, depending on what's going to happen in the next 6 months.
And then we have antivirals. And fortunately, both of our commonly used antivirals, both the neuraminidase inhibitor, oseltamivir, and baloxavir, the new antiviral, seem to be effective. The worry with oseltamivir is that we know with that antiviral, you can get resistance because of the N1 reasonably quickly. We know that we do have a backup with baloxavir, but again, we have oseltamivir in the national stockpile, which, as long as the virus remains susceptible, will be very good and really needs to be thought about right now in terms of trying to prevent transmission, because the way we're going to get reassortment or mutation is through continued transmission. And that's one of the things we really need to look at to try to cut down on the amount of number of transmission events that are occurring within humans.
Can I ask you a question, Dr. Monto, about treatment? So a lot of people are very interested in whether nirmatrelvir, the half of Paxlovid, might be active against these viruses, because we got a lot of Paxlovid in stockpile. So do you have any thoughts about that? People are looking.
I really don't know very much about that. It would be relatively easy to find out. So instead of speculating, of some very simple experiments could really give us the answer on that one.
Given what you're saying now, I wonder if I can put a slightly more positive spin on everything. The good news right now is that while we have this large outbreak in cattle, we don't have so many human infections. We certainly don't have an outbreak of serious illness in humans. And the fact that there's so much animal disease out there, and that there really is an imminent threat, and it's appearing in the US where it's right in front of our noses, do you think this is the spur to do things that we have needed to do all along? Arnold, you mentioned developing the broad range vaccine. Jeanne, you mentioned the BNABs, the broadly neutralizing antibodies. Is this the time?
Well, I wonder whether this is the first time we've had this occur. If there hadn't been a few things happening, we wouldn't have detected that this was just low milk production in dairy cattle. Since this is a global pathogen, if you're having human cases in Cambodia, I can't believe there haven't been cases that have been missed in dairy cattle or in cattle. That's the other thing. All the attention is being given to dairy cattle. There's no difference in terms of receptors in beef cattle, and they're often on the same farms. And everything, all of these domestic animals are shipped around the country. A lot of work has been done to follow swine influenza, and the pigs are shipped from North Carolina to Iowa because it's easier to ship the cattle than to ship the feed that they're given, and for other economic reasons. So we're dealing right now with a situation where we need both more information, more data, more concern about the problem, but a realization that this virus has been trying to get into humans and to spread in humans for at least 25 years, and it hasn't made it yet. We even had, in the 2005 period, human infections from backyard flocks in Egypt and Southeast Asia, and cases didn't even spread when there were household infections. And the hypothesis, never proven, was that the receptors may be different in different individuals because blood relations tend to get transmission, but not others in the same household. So we really don't understand a lot of this, but to agree with you, we shouldn't be panicked by this. And we need to remember that a wake-up call is only useful if you do something about it while you're awake.
I like how you took my positive question and you turned it so negative, Arnold. Thanks.
No, I still think I still think this is low risk, but it's but we've got to be realistic that it is high high risk if something does happen.
So, any last things that Lindsey? Any last questions or anything last things that people want to say?
In listening to your comments, Jeanne and Arnold, what I hear concern because there's widespread outbreak in a new mammalian species. However, this organism has been trying to do this for decades, so hopefully there is some species barrier. What I also hear is the incredible advances that an investment in science has allowed through NIH, through CDC, through other agencies and industry partners, from the understanding the molecular biology of influenza and its segments and how the receptors behave, to the development of vaccine constructs and platforms that can have swaps or be potentially re-engineered quickly, to the BNAB technology, which can be more broadly applied, and then antivirals that may well work across different flu A's, depending on the reassortment. So I see this as, on the one hand, we shouldn't diminish the concern of this threat. On the other hand, we are in a much better place now than 20 years ago through the scientific investment, and to use this as an opportunity to invest more in understanding the biology so countermeasures can be deployed more rapidly to protect our communities. That's the message I'm hearing, which to me is very encouraging, but we still have to stay very vigilant.
I certainly agree.
I would agree as well. The only other thing I would add, this is not a negative, but just something to remind folks, we've been talking about One Health for a long time, and that is human health and animal health, particularly in this agricultural context. It's time to re-examine that, I think, as a commitment to really try to get a sense on what's happening here in the surveillance. Because as Arnold said, if we don't know what's going on, we can't really be awake for it. And it's really critical that we're awake for this as it evolves.
And the reason One Health was developed was because of the barriers and understanding that anything that goes on in non-human species is of concern. And we're not there yet.
Thank you, Arnold. Thank you, Jeanne, for joining us today. And thanks, Eric. Thanks, Lindsey.